our third set of notes, which is on meiosis II. After watching the first video set, you realize that meiosis breaks into two sets, meiosis I and meiosis II. You've also spent some time looking at your textbook that you can see that there's a difference between the two of them. But yet there's a lot of similarities. It seems like the same thing is going on. Well, they are pretty close to each other. They both go through their PMAT. There's just a couple of differences. First of all, one of the differences is the end result. So at the end of meiosis I, you had 2 cells. At the end of meiosis II, you end up with 4 cells. Another difference that you're going to look at is how many cells are going through this at the same time. So for meiosis I, you had 1 cell going through the process, and at the end formed these 2 cells. Well, in meiosis II, these 2 cells then are continuing through the process of PMAT, and you end up with a total of 4 cells. Now, there's a difference between the chromosomes, and we'll look at that towards the end of this slide. So one big difference that we have between meiosis I and meiosis II is how we start. At the beginning of meiosis I, in prophase, we had a diploid cell. It had both sets of chromosomes. In fact, 46 total chromosomes. At the end of meiosis I, we ended up with 2 haploid cells. Each cell, remember I think of haploid as have half the number of chromosomes as the original cell. So prophase is going to start now, prophase II, with a haploid cell. We're going to continue to talk about prophase II just a little bit more. And the steps are the same things that we've had back with PMAT with mitosis. The chromosomes will become visible, the nuclear membrane dissolves, and the spindle fibers form. And what you can see is that I have a cell off to my right that has 2 chromosomes. They're non-homologous chromosomes. And those non-homologous chromosomes will start to appear. We are now on the M for our PMAT, which is metaphase, and we're looking at metaphase II. And it's the same steps. Chromosomes move to the equator, they line up, or I say they meet in the middle. At this point, the spindles will attach to the kinetochores. Remember, that's that particle in the centromere that they actually attach to. Now, one thing I want to make sure that you understand that is way different in this between mitosis and meiosis II, because mitosis and meiosis II look the same. The only difference we have between those two is the number of chromosomes. In metaphase II, in meiosis, I have 23 total chromosomes that are lined up, 23 X's. And if we go back to mitosis, there were 46 chromosomes that lined up in a row. So right away, you're seeing a difference in the total number of chromosomes. In fact, in mitosis, it doesn't matter with homologous pairs. They do not line up. They will have all 46. It doesn't matter what order they line up either. Whereas in meiosis, it does matter. The homologous chromosomes matter of where they line up. So this will look a lot like mitosis, but they are not exactly the same because of the number of chromosomes. Okay, time for our A in PMAT, which is anaphase II. And if you remember from last chapter, I told you that anaphase, the A means away. And at this point, our centromeres will divide. Remember, that's the part that holds the X's together. The spindle fibers will shorten. And those spindle fibers will pull each of the chromatid strands to opposite poles. The T in PMAT stands for telophase. Or I kind of like to think of 2, but 2 is not working in this case. If we look at back in mitosis, we form 2 cells. And after the end of telophase in meiosis I, we form 2 cells. This is the different telophase. Because of the fact that I started with 2 cells simultaneously going through this, I'm going to end up with a total of 4 cells. So this is the telophase that is different than the other 2 because of the fact that we have 4 cells. I also am going to have 4 nuclear membrane that will separate these DNA strands. These chromosomes are going to start uncoiling, the spindles are going to disappear, and it's going to start looking like a normal cell again. As soon as we are done with PMAT, remember that we get into cytokinesis right away, which is the separating of the cell membrane. So we're going to have the cell pinch in half. That cell membrane is going to elongate to wrap around stuff. And then we have an ending of 4 total cells. Oh no, a slide with so much information. Actually, my point of going through the slide is just to relate what's going on in each of mitosis, meiosis I and meiosis II. And also for you to understand that even though meiosis II looks like mitosis, there's some differences between the two of them. So let's go over mitosis. And one thing I want to make sure you understand is that during that S phase, remember synthesis phase and interphase, it will duplicate those chromosomes. And so it starts off with 46 chromosomes with 92 chromatids. Now, when mitosis goes through PMAT, all 46 will line up in the middle. They're not buddy-buddy or anything like that. They're going to line up together. And as it gets separated, I end up with 46 chromosomes with 46 chromatids in the daughter cells. That means that in this case, a chromatid is a chromosome. Again, a little confusion between definition. Chromatids are basically counting the centromeres. And remember that sometimes you can have a centromere and an X, and sometimes when the X's separate, each of those separate ones have a centromere. Chromosomes are just basically condensed DNA. So chromosomes can either have 1 chromatid or 2 chromatids by definition. And I kind of have that in the yellow towards the end, trying to refresh your mind between those two things. So it is a little confusing understanding the difference between chromosomes and chromatids. Now, remember that we don't have that full set of chromosomes. That happens. Then the next time the cell decides to split, in the S phase, everything's going to get duplicated all over again. All right, now let's go on to meiosis I. We start out with 46 chromosomes with 92 chromatids, just like mitosis. The difference between mitosis and meiosis I is that when we line up for metaphase, we're lining up with our buddies, our homologous chromosome pair. Except for gentlemen, if your 23rd chromosome is an X and Y, those two are stuck together even though they don't want to be because they don't see each other as identical. We kind of looked at that in the video the other day, and that's why they don't cross over. Whereas ladies, your 23 chromosomes are both X's. They're going to be lined up like the rest of them, no different. So we end up with a line 2 by 2, 23 2 by 2s down the middle of metaphase. When they separate, we end up with a total of 23 chromosomes and 46 chromatids. There was no separating of the X's, so each chromosome has 2 chromatids. Now let's go on to the yellow, the meiosis II. Meiosis II looks identical to mitosis. The only difference is how many chromosomes we have go through it. Mitosis has 46. Meiosis II only has 23. So meiosis II, we start with 23 chromosomes, 23 X's. And because they're in the X shape, then there's a chromatid on either side. It will end with 23 chromosomes with 23 chromatids. So when we separate at the end, the X's are separated. So we end up with 23 half X's, if that makes sense. And then remember how I told you before, a chromosome may have 1 or 2 chromatids. And that's kind of a way to kind of figure that out or understand this a little bit more. So at the end here, I wanted to end with a picture. The yellow is going through meiosis I, meiosis II. So the steps of meiosis. We start off with a diploid cell. We end up with 4 haploids. You can see there's little brackets. The top first bracket on the left signifies meiosis I. The other bracket to the right and shows what's going on in meiosis II. On the bottom in the pink, you can see that I have just regular mitosis. Mitosis starts with a diploid cell and ends up with 2 diploid cells at the end. That's it for the set of notes.